but consistently. Okay, so I think we can start. So welcome again to the spring college, which is clearly not in spring. So I'll be teaching for 2 weeks, as you know from the program, and what I'll be teaching is divided into quite disconnected parts. So the first part is on quantitative microbial physiology, and today is sort of an introduction to that. And the second part is more on critical thinking in the era of big data. So roughly, I will not tell anything about the second part because there is going to be time in the second part. Roughly, we will transition around Monday, Tuesday next week. So this first part, it's longer than the second part. Yes, I think we can start with that. And my idea is also that this first part I can sort of gauge what is most interesting for you during sort of the second half of this week, and so there is some flexibility in what I'm going to cover. So, but today is going to be a very gentle introduction in microbial physiology, which is almost like a high school kind of summary of what you should remember, but none of us will remember. Okay. So first of all, this first part is on, as I said, microbial physiology. I'll explain what physiology is. But first of all, let's talk about why we care about microbes. So this is one of the most recent version of the tree of life that we know. So the way this is built is basically you take an important component of every living things, which are ribosomes, and I'll remember what ribosomes are in the course of the lecture. You take ribosomes, you look how they are made, and you compare them. And so you say, since this is something that all living forms have, if two ribosomes of two different organisms are similar, they have a common ancestor which is very close in time. If it's very, very different, if it becomes more and more different, you expect this ancestor to be much back in time in a distant past. And so here is what you obtain. And there are three main branches. So these are eukaryotes. These are archaea, which are unicellular organisms, which have, as a first approximation, you can imagine that are something like in between bacteria and unicellular eukaryotes. And these are bacteria. So the first things you notice is that we are here. Okay? And here there is us, fishes in the sea, mushrooms you can eat. All eukaryotes are here. So most of life, except a part of this part here, most of life is unicellular, and most of life is actually bacteria. So the spread, the fact that you have so much spread in this tree means that actually the level of diversification of these organisms is very, very high. So there are organisms that have completely different strategy and populate all the corners of our spherical planet. Okay? Now, another interesting thing, which I'll not go through, but it's like interesting, is that you will see dots which are branches that have a red dot. These red dots are organisms that we are not able to grow in the lab. So these are things that are out there, but that we are not able to grow, and basically they were inferred by reconstructing parts of their genome collected from the environment. So what is also interesting, and I will not go through that, is that a vast part of this diversity is something that we cannot reproduce in the lab. When we try to grow these things, to isolate them, they don't grow. We're not able to grow them. Okay? Questions?
But they are found in nature.
They are found in nature. We know that they exist. For instance, it's a very interesting story. So these things here are very small organisms, which are probably parasitic, and you see that sort of it's a big, it's quite separated for the rest. So it's a big radiation that happened many millions, if not billions years ago. And we are not able to culture any of them, but we know that they exist. I mean, people saw them under the microscope, but we cannot grow them. Yes?
Sorry, do the positions of each species here matter? Is this like a PCA thing? Or just
The position in space doesn't matter. What matters are the relative distances. Now here there is a big approximation, which is the fact that somehow the way this is constructed is that you take a lot of sequences of these ribosomes, and then what you build is you assume that these sequences come from a process where there is differentiation, right? So there is underlying a tree, a network which is a tree, and things differentiate right, this way, and you try to infer what is the most probable tree. Which is a very different thing to say, I mean, this correlates with what is the clonal tree, so when there was the common ancestor, right? But it's a different thing for multiple reasons, okay? Other questions? Something that I didn't say, but it was not needed. Please stop me anytime, ask questions, etc., etc. Okay, so as I said, there is a lot of diversity, and the result of this very large diversity is that microbes are really everywhere. So these are some extreme cases. So this one, it's an archaea that, so it's a microbe, a unicellular organism that lives like in hydrothermal vents, so in the depth of the sea, at very high pressure. And this organism has its optimal growth condition above 100 degrees Celsius. This is extreme pressures, and it grows pretty fast at these extreme conditions. This is another organisms that lives in hot springs at extremely low pH, so extremely acidic condition, and again, can reproduce quite rapidly, so it duplicates, a cell becomes 2 cells in about 6 hours. And it's a very interesting story. So basically, there was a way to remove like bacteria and other contaminants from beef, which was to irradiate with some doses of ionizing radiation, so to kill everything that was inside. And people found out that this kind of meat that was irradiated got spoiled by some bugs. So they isolated these, and they found these bacteria that can survive extreme doses of radiation. And this guy, it's like basically, I don't know, a super bacteria in the sense that can survive extreme cold, dehydration, survive 3 years in outer space. So you can do whatever, and then you put it back and can grow again. So this is to say that these organisms are really able to extreme adaptation, so they are able to live and survive and grow happily in very, very extreme conditions. The other point is that, well, I don't know how much you care about this, but also these microbes are extremely important for many processes that occur on our planet. So they are essential for biogeochemical cycles. For instance, nitrogen fixation. So there is biogeochemical cycles of nutrients, and these biogeochemical cycles, so for instance, how nitrogen is fixed in soils and then can be absorbed by plants can only be done by microbes. Similarly, you know that we have microbes and bacteria living in our body. So an interesting thing is that for every human cell that you have, there are 4 bacteria in your body. And it's about 1.5 kilograms. And they perform essential functions. So for instance, they synthesize essential vitamins like vitamin K2, and they regulate our digestive process in the gut, but also they regulate many things, for instance, on our skin, etc. So basically, they are an essential component of life on our planet. Okay. Now, what we're gonna do in these few days is try to understand something about bacteria, and in doing so, we will forget all this diversity. We'll focus mainly on one bacteria with the idea that these prototypical species will tell us something about how they function. And in particular, the main question that we want to ask is, we have these simple, apparently simple organisms, bacteria, but we saw that they are many diverse, they can do many adaptations. And the question is, what do they have to do to be alive? So basically, what are the functions that they have to perform to be alive? And in particular, what we will try to do is to do something quantitatively. And in general, a big branch of biology is physiology, which studies basically all the things that occur inside a cell to keep the cell alive. What we're going to try to do is to study this in a quantitative way. So put numbers and write equations and make prediction of these equations to study what are the fundamental processes that occur inside a cell that make this cell being a living thing. Okay? So our goal is to, I'll teach you sort of some recent results which combine theory and experiments to really develop a theory of how these bacteria work. Yes?
You haven't mentioned probably viruses. So
yes.
Bacteria is something else than viruses?
Yes. So.
Cell also?
So viruses are depends how much you like definitions, but are at the edge of what is living and non-living. So the difference between, so they are much smaller, first of all. So a typical bacteria is a few micrometers. Virus, it's like tens, hundreds of, with some exceptions, tens, hundreds of nanometers. And viruses are, I said they are at the edge of what is living and non-living because they are not able to perform metabolism by themselves, and they are not able to reproduce by themselves. So what viruses do is that they have only genetic material, and they attach to some cell, which can be a bacterial cell, can be our cells. They inject their genetic material inside the cell, and then they use the machinery that I will describe later to produce other viruses. But a virus itself is not able to get energy from the environment, is not able to reproduce by itself. It needs another living form to do all these functions. Okay? Other questions? Okay. So my final secret and not so secret goal in teaching you this is that I want to communicate a few things. So one is that, microbes, as an example of biological systems, do very interesting and extremely complex things, extremely complex tasks. So even if most of you, when I really didn't like biology when I started studying physics, but now I really enjoy it, and I think it's really fun. There are a lot of things to learn and a lot of things that physicists can do. And so this is my main message and the main things I want to communicate. And in particular, I want to show you that even in biology, even if these things that move and do stuff are extremely complex, it's possible to find quantitative laws. So patterns that are very reproducible and from which that sort of constrains how this system behave. And that it's possible to build quantitative predictive theory in biology. So it's possible to write down equations, and then with these equations derive predictions, and then test this prediction quantitatively in biology. And when I say biology, I want to specify that I really mean how biological system works, right? Obviously, you can take E. coli, a bacteria, and throw it out of the Tower of Pisa, and it's going to fall using like sort of the same as any other animate or inanimate object, but that's not biology, right? What I want to convince you is really the way this organism functions can be understood using math, if you want, or can be understood in quantitative terms. Okay, so this is my goal. Questions? Very nice. So what I want to do today is to do a little summary of the few things you need to know or remember about biology. So is there any biologist in the room? Trained biologist? Okay, so I can say whatever I want. No, I'm joking. Okay, so this is like a short summary in basically 5 slides of the things you need to recollect to understand whatever I'm going to say in the next. And just for reference, there is this book, which is a sort of, I don't know, standard book where everything is written about microorganisms, which is like, I don't know, 1,500 pages. And what I'm covering now, it's in about 150 pages. So I'll show you a very condensed part of it. Okay? Good. So I need also these things, if it opens. Okay. So the first part, which is actually not about biology, but about chemistry, is that these things doesn't open, doesn't recognize my face. Now it does. Okay, so the first thing is that there exist chemical reactions, right? We are probably all aware of these. And so chemical reactions, what they do is that they proceed. So you have some reactants, and with some rate, there are some products. And what you probably know is that this rate sort of depends on some, well, there is a constant rate because basically in transforming these reactants to products, there is some energy barrier to cross. And so basically there is the rate at which you transform reactant to products depends on some activation energy and temperature. Okay? Now, what you probably expect is that when you have one reaction in one verse, you have also the reverse reaction, which is possible. And if you wait a long time, what do you think is going to happen? Exactly. So if you wait a long time, you're going to go in chemical equilibrium, which means that basically the rate at which the total rate at which this reaction occurs is the same as the reaction back. So if we, for instance, we track the concentration of the reactant and the concentration of products, so, and we write what is chemical equilibrium, what we have is that the rate, total rate at which you move to the left in this reaction is going to be to the opposite. okay? And so in chemical equilibrium, what you have is that this condition holds. So if you want, well, okay. So you will have that this simple condition applies. Now, an important point, which I don't know how obvious it is that cells, living organisms, are intrinsically out of this equilibrium. So they are never in chemical equilibrium. And the reason why they are not in chemical equilibrium is because what a cell does is that it has a membrane, so it keeps the out being different from keep an outside and an inside. And what a cell constantly does is that it keeps getting things from the environment actively and pushing things out of the cell. Okay? And so maintaining this imbalance between the external world, what is outside and what is inside, is what maintains cells out of chemical equilibrium. Okay? Questions about this? Good. Now, the second point about chemical reactions is that there are some chemical reactions that, as a net effect, release energy, and some chemical reactions that needs energy to occur. Okay? Now, cells perform both the reaction act. Yes? Mm-hmm. Okay, so the point is that a cell, first things that you need to know about a cell is that it has a membrane, okay? So it has an inside and it has an outside, okay? So a cell performs a lot of reactions inside, so transforms something into something else into something else. If you just take something which is a closed system and you start performing on this reaction, at the end, if you wait a long enough time, you're going to go to chemical equilibrium. Okay? The reason why, and if this membrane was just something passive where things could diffuse in and diffuse out, passively a cell will become in chemical equilibrium with outside. Right? The key point is that what these cells do is that they actively import things from the environment, and they actively export things outside. Okay? So the composition of a cell is radically different from what is outside the cell. Okay? And this constant battle of a cell to keep its inside different from the outside is what maintains the cell out of chemical equilibrium. Okay? So this is to say from modeling perspective is that we'll never think about chemical equilibrium in the models we're going to write, right? We'll never think that we have a cell, and then we wait a long time, and then everything is in equilibrium, right? Because if you want, first characteristic about a living cell is that it's out of this equilibrium. Yes, question?
So what is the difference between reactants and products? Reactants are inside the cell?
No, this is just because this is just, I'm thinking of looking at something that reacts and becomes something else. But of course, if you reverse it, there is no difference. Okay?
So
I'm just.
Is the left-hand side, the S.
Yeah, could be the right-hand side. Yeah, there is a symmetry in what can occur. There is an asymmetry in what is the rate at which the reaction occurs.
And if there is nothing that goes inside the cell, then it's a stable system.
If the cell, if you wait a long time and the cell doesn't perform any action to push things outside, push things inside, then it goes to chemical equilibrium. Right?
And where is that on the graph?
There is nothing on the graph. On the graph, what this shows you is that if you start with some reactants, you start with some product, you have two types of reactions that release energy as a output of the reaction, and reactions that needs energy to be performed. Okay? Now, the point I wanted to make with these things about energy is that organisms do both type of reactions. So they both do reactions that release energy by transforming some substrate. So they take some material, and then basically, because there are some chemical reactions, some energy is released, okay? So metabolism, so in general, the set of chemical reactions that cell performs can be divided in two, which is a sort of, in some sense, artificial distinction, but roughly can be divided in two, saying that there are reactions that are part of catabolism, so that they release energy, so they take some material, and the reactions of some material produce energy plus other material, byproducts. And there is also, cells also do biosynthesis, so they produce new material, right? So they use energy and some substrate to produce more complex, typically, material, okay? Now, of course, as a caveat, this is not the only way to produce energy. Another way is to do photosynthesis and use light to produce energy. Okay? Questions about this? Okay. So what we learned so far is that there are, inside the cells, there are chemical reactions, and this chemical reaction can be used to produce energy or to produce new material. Now, yes?
These two ways, by material or by light, are just the main one. But there is like by heat, for example, some microbes produce energy from heat, like, in the volcanic activity in the sea, the microbes that live there.
Well, but they use, no, typically they use some chemicals to do this. Yes, yes. There is a big distinction on, well, yeah. They do, yes, but, okay. Good. Now, the point is, there are reactions. Okay, very nice. Now, these reactions are not something, I mean, let's say, the cells have a say or can tune how fast the reactions are. And the way cells tune how fast the reactions are is by using enzymes. So what are enzymes? Enzymes are proteins that, by binding to reactants, decrease the activation energy of a reaction, and so they speed up the rate at which a reaction occurs. Okay? So, imagine that you have, for instance, that we are out of equilibrium, and we don't have any product, and we have this reaction occurring, and sort of we forget about the reverse reaction. So I'll write here, otherwise you will not see. So let's say that we focus on what is the concentration of the product, and we say that K-rev is equal to 0. Okay? Now, what we have is that we have the concentration of the reactants, of the substrate, and there is some rate at which this occurs. So this is what happens if you have like sort of spontaneous, a reaction spontaneously occurring. The key point of this is, basically this is a translation of this reaction. The key point of this is that you see that the accumulation of the products is linear on the substrate. Right? So we start from something, and the rate at which you produce whatever you have, it's linear in what you started from. Okay? The more there is, the faster it is. Now, what I want to show you is that if you have, well, I want to show you how enzyme enters into this, but I also want to show you that enzyme entering into this can sort of modify radically how this occurs. So what does an enzyme do? So this is like S2P. So what an enzyme does is that you start from S and an enzyme. Okay? And what they do is that they bind into some unit, and then you get a product. Okay? Now, one important aspect, which is sort of pictured here, is that by doing so, what an enzyme does is that it can reduce this energy, so can make this reaction occurring faster. Okay? I want to show you that this is not the only outcome. There is also another outcome which becomes very, very important. So here we have two rates. So one is a binding rate, so the rate at which the enzyme binds to the substrate. And the other one is what is called the catalysis rate, so the rate at which the reaction occurs once the enzyme is bound to the substrate. Okay? Do you see back there? It's too small. It's okay? Good. So now what we have to write is we have to write basically two equations, right? So what we have is that we have the concentration of products, which will be equal to the catalysis rate times the concentration of the enzyme bound to the substrate. Okay. And then what we have to write is what is the rate at which how does the concentration of bound enzymes change in time? And so the idea is that this change in time, because we have Kb times E times S, okay? So the rate at which this is produced is the binding rate times the product of the concentration, okay? And then we have the catalysis, which remove these and produce new enzymes. So this is Kcat. Yes. This one? Ah, sorry, yes. Yes. It's just the join, yes. Thanks. Okay? Now, what we can do is that we can say, well, these reactions will have these two terms, so if we wait a large enough time, this will become equal to 0. Or if you want, these two rates balance. And what we get if we set this equal to 0 is that we get that ES is equal to Kcat ES is equal to Kb E times S. Okay? Now, what we can say is that, well, we will have total concentration of enzymes, which is given by E and the ones that are bound. Okay? So this is the total amount of enzymes that we have. So if here we write E dot minus ES, what we get is that ES is equal to E dot times Kb S over Kb S plus Kcat. Okay? Do you agree? So just to summarize what I did. So I said the rate at which you produce product now is equal to the second step, so it's equal to the rate at which catalysis occurs when the enzyme is bound to the substrate. But then how many enzymes bound to the substrate I have? I have two processes. One is the rate at which a free enzyme binds to the substrate, and the other one is when the catalysis occurs, okay, when they unbind. Then I said, well, I'm keeping the total number of enzymes fixed, so I have this sum, and then I set this equation to 0. I substitute the free enzymes as the total minus the ones that are bound. I solve and I get this expression, okay? Okay, and so what I get is that now, if I substitute this expression in the first equation, I can substitute it here, what I get is k cat e dot, and I can write this as S over S plus k cat over kb. So what we started from is that we had dp dt equal to kp, but now what we have is dp dt equal to, sorry, here was S, equal to something else, which is some total, well, let's say some parameter V max, S over S plus Km, okay? Where this V max and Km are equal to this term here. Okay? Now this is fundamentally different from before, because what we had before is that we had that the rate at which the product was produced as a function of the substrate was something linear. Okay? While now what we have is that we have something with a sigmoidal shape, right? So something which is still, well, linear at the beginning and then saturates, right? And this value here, it's Km, and this value here, it's V max. Okay? Now, why is this more interesting than before? Yes, sorry, yes. Sorry, it should be like this. Yes. Why is this more interesting than before? Yes, yes, because it saturates at some point. It's interesting because, well, it saturates at some point, but also because now there are two parameters, right, which tell us how fast is the reaction when it saturates, and another parameter that tells us when the reaction starts to saturate. So what is the scale of the product? And now these two parameters are related to what? Are related to how many enzymes we have, okay, and what are these two rates? Okay. Now these are fundamentally, I mean, what are the rates are fundamentally chemical properties of the enzymes, which tells us, for instance, what is the affinity of the enzyme to bind to the substrate, right? Or what is the catalysis rate, right? So by changing the structure of the enzymes, not by changing actively, but the structure of the enzymes determines basically how fast effectively you can produce and what is the dependency of the production rate on the concentration of the substrate, right? So you can somehow tune the output based on the input, okay? Yes.
The K probability is a constant number, or it for each substance and each enzyme?
Yes.
It is a constant, or
Yes.
it change depending on something else?
So I mean, it depends on, when you say constant, it depends on the time scale, right? So it's a constant on the time scale of what we care now, which is about the physiology, so the reaction occurring while a cell is growing. It's not constant on the evolutionary terms, in the sense that you can change, I mean, not you, the enzyme, yes, the enzyme, sorry, I'm putting agency where there should not be. So that the enzyme can change, and so this mapping can change.
In the reaction itself, it is constant, or
Yes, 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 yes. And now you can make, one of the reasons why I plotted the wrong one is because what you can do is that you can actually make reaction more complex. So you can add multiple steps where you can have to, basically you have cooperative actions where you can bind multiple enzymes or multiple things, and then you can you can have different shapes here, okay? And so you can basically make the production of something depending on the substrate in multiple ways. Okay? Yes. This one is this one, right? I just wrote this Vmax, okay, and this Km, okay? Okay? Other questions? Yes.
Multiple products can be made by one substrate, or?
Yeah, I mean, here I'm sort of hugely simplifying this in the sense that I'm considering that there is one substance making one product, but in reality you have multiple multiple reactants making multiple products. So I mean, what matters is the concentration of all of them and the product of their concentration. Yes.
In the other case, we can do the same method to...
Yes, exactly. You can, I mean, this is like sort of the simplest version, but you can imagine of writing these with more steps, and then you can derive exactly the same conditions. Okay? Yes, so basically you write these as the final step, and then you assume that all the intermediate steps are at equilibrium. Okay? At equilibrium in the sense that there is something coming in, something coming out, and they're balanced. Okay? Other questions? Yes.
Yes, sorry, so all enzymes will help us to get products?
So what the enzymes do, the first things that they do is that they can tune the speed of a reaction. So a reaction that will take a long time, they can decrease this activation barrier. So they can change the way the reaction occurs, making it occurring much faster. Okay? So there are reactions that will never occur in the lifetime of an organism that are made possible because there are enzymes. Okay?
So is there any enzyme that doesn't work for this process? Like in the equation, can we have any enzyme that doesn't help to make the process faster?
No, the point is, okay, so the point is in the second part of the title that not only enzymes exist, but they are also specific. So they catalyze specific reactions. So you have one reaction, so transforming some products into something else. That reaction has a specific enzyme. Okay? So when you have multiple reactions, you have one enzyme for each of these reactions. Okay? So they are specific. It's not that you have something that is a general and speed up everything. Right? Yes?
So are these equations a closed system or organism where the temperature changes, so K could depend on the temperature?
Yes, I mean, K depends on the temp. I mean, all these parameters depend on temperature, yes. Now, what I'm considering is that these conditions are fixed. Okay, then of course you can make things more complicated.
So it's isolated system?
Yes, for now, yes. Other questions? Okay. There was a question. Yes. Hmm? Sorry, can you speak a bit louder? The physical interpretation of this one? This one? This one? So this is what this is telling you, how the concentration of enzymes bound to the substrate depends on the concentration of the substrate. Okay? Yes. Yes, because I got confused, because you can make this process more complicated, like with multiple steps, for instance, when you need to have multiple components coming together. And if you do that, what you find is not something like this, but something where these are raised to some exponent. Okay? And so in that case, you can basically tune how fast the reaction occurs as function of you can make these more like a sigmoid curve. Okay? Other questions? Yes. How they, so basically, intuitively, what happens is that if you have these enzymes, there are two regimes. So there is one regime, which is the initial one, which is the same as before, right? Where the reaction basically is limited by how much this process is fast. Okay? And then you have another regime, in this case, which is limited by how many enzymes you have, okay? Which is this second step. Okay? So there is a moment where you don't have enough, even if you have very, very large substrate, right? Given that this reaction has to be catalyzed by an enzyme, you don't have enough enzyme to make it occur fast enough. The point is, mathematically, you just solve this, and you see that here you get this ratio, which is the ratio of these two terms, which determines where this transition occurs. Okay? Other questions? Okay. Good. So there is another, okay, yes. Yes.
And then when we develop it and we were able to
Yeah.
and it's not linear.
Yes.
We have two phases, the linear one
And then saturates, yes. Okay. Now, the point out of this is there are reactions. Reactions, they need enzymes to occur because they can sort of lower this activation barrier, and by existing enzymes can make can make a reaction occurring more or less rapidly, right? You see that the saturation point depends on how many enzymes you have. Right? And what I told you is that enzymes are specific. So an enzyme corresponds to one small set of reactions, one or let's say one reaction. Okay? Now the second point is, well, for now it seems disconnected, but the instructions to make every proteins, including enzymes, are written in DNA, and they are and they occur in a two-step process. So we said there are enzymes, and these enzymes are needed to have reaction occurrence. The other point is the instruction on where to build these enzymes are written in DNA, and there are two steps in which they are built. So transcription and translation, okay? So I'm really going back to high school kind of material. Now, these two steps, let's look at them individually. So the first point is you have DNA, okay? You in an abstract sense, but also you have DNA. On DNA, it's written the instructions to make these proteins. So what happens is that there is one thing one machinery which is called RNA polymerase, which attach to DNA and start writing some strand of RNA, okay, which you can imagine just as a linear chain with some instruction written, right? You start writing that, and after this process, what you have is that you have this, what is called messenger RNA, which is around in the cell, okay? This is the first things which is called transcription, okay? Everybody on page? Yes. The second step is what is called translation. So basically, what happens is that there is another machinery, which will become very important for what we're going to do. So remember it, which are called ribosomes, which are very big fatty proteins that take this messenger RNA, so this linear strand with instructions, read it, and as they read it, they assemble amino acids. So there are amino acids which are the building blocks of proteins, and as they read this messenger RNA strand, they start attaching the right amino acids corresponds to the sequence they read, okay? And so they start doing this, and they basically what they do is that they assemble a sequence of amino acids. At a certain point, they finish to assemble this, and they have a sequence of amino acid. And then what this sequence of amino acid does is that it folds and produces a protein or a structure of a part of a protein. Okay? So I see, while I was explaining this, there were some phrases that were saying, yes, I know this, I saw it so many times, you are so boring. And others, they say I'm completely lost. So what I'm going to do is that I repeat it again. Okay? So let me repeat it again, so then I hope everyone is very bored, and then, okay? So we have DNA, right? On DNA, there are instructions, okay? We don't need to know about how they're written for now. We have instructions. Now, there is one thing, which is a machinery, called RNA polymerase, that attach to DNA and starts reading these instructions. It reads the instruction and makes a copy of them. Okay? It's not an exact copy, but let's say it's a copy of them. So it makes a copy, and this copy is this orange thingy, which is messenger RNA. This messenger RNA goes happily around in the cell, and sometimes it encounters a ribosomes, this big protein. What this protein does is that it attaches to the mRNA and starts sliding, or actually the mRNA slides through it, and then what it does is that it finds the right amino acids corresponding to the right sequence of letters in mRNA, okay? And so when it finds it, it's sort of attached, and it creates a sequence of amino acids, okay? And then when it does, this sequence of amino acids folds and becomes a protein. Is it clear? Now, more people were bored by my second one, so it's a success. Questions about this? So this is basically how everything inside a cell, every protein inside a cell is made, okay? So this is how proteins, and so in particular enzymes, so the things that make reaction occur, are made inside a cell, okay? So for now, I told you two pieces of what is important, and now I'm putting them together. So the first thing I told you is that reaction in a cell need enzyme to occur, and so enzyme can tune how fast reaction occurs, okay? And then I told you that the instructions to make these enzymes are written in DNA, so cells know how to make these enzymes, and can be produced in this two-step process, okay? Now the key point is, now how does a cell decide how many enzymes produce of a given type, right? So imagine that you are a cell, you have cells, but now do a very things of introspection and imagine you are a single cell. And I mean, actually, no, you can imagine that you want to do what you're doing now. So now, in 35 minutes, we're going to be upstairs and start having lunch. And so you need to start producing enzymes to digest the food you ate. Right? So how are you going to produce these enzymes? Right? The instructions are written in DNA, so you know how to do them. But you have to decide, okay, I'm going to produce more. Right? What I told you so far is, okay, RNA polymerase binds to DNA, and then you start transcribing the messenger RNA, and then ribosomes translate the messenger RNA into proteins, etc. So there is a way to make them. But now, if the condition change, so now you start eating, how do you decide to do that? How do you change the fact that sometimes you produce, sometimes you don't produce them? Is the question clear? Okay. Okay. The answer is regulation. So what I told you so far is that you have some region on the DNA, and then RNA polymerase attach to DNA and start transcribing. Okay? So it starts reading the instructions written on DNA and producing this messenger RNA, okay? That then serves to produce proteins. Now, a missing part of what I told you is that there are other proteins, which are called transcription factors, which can bind to DNA. Okay? So for instance, this is a simple way this occurs. So you have a protein, which is called a repressor. Okay? So this protein here, if it's unbound to the DNA, the RNA polymerase can proceed happily and produce the mRNA that serves to produce the enzyme. Now, in this case, if there is some other molecule around, in this case is an amino acid arginine, if arginine is bound to this protein, okay, what happens is that this repressor changes conformation and attach to DNA, blocking the RNA polymerase going around. Okay? I repeat once more. So you have this protein going around, and there is no arginine. Okay? There is no arginine, and this protein is going around and is doing nothing. Now there is arginine. Okay? There is arginine. Arginine binds to these proteins, to this repressor, and when it's bound, this repressor came conformation and now wants to attach to DNA. It attached to DNA, and RNA polymerase cannot move, and it doesn't produce the mRNA. Okay? Yes?
So the kind of activation function of this repressor protein is dependent on the presence of arginine?
Yes. So we're going to write down an equation for this if you have time today. But the point is the presence of arginine. So you can imagine that this protein can be in two states, bound to arginine or unbound. If it's bound to arginine,
What triggers the presence of arginine?
The beautiful thing is that what this does, it produces arginine. Okay? So this is an example where you have a feedback loop. Right? There is no arginine around. You produce arginine. I mean, this could be whatever. So in this particular example, there is a feedback loop. So there is no arginine around. You start produce arginine. Arginine accumulates. Arginine bounds to this. You stop producing arginine. Okay? So in this case, you want to produce arginine, but you don't just want to produce it like indefinitely, continuously. You want to arrive to some level, and we're going to see what this level depends on. But you want to arrive to some level, and so you have these mechanisms that regulates, in this case, these proteins that serves to produce arginine.
What if we want to produce another protein?
If you want to produce another protein, what you have is that this is a piece of DNA, then you have another piece of DNA. Let's say you are now, this morning, some people at breakfast were talking about lactose intolerance. And, no, that was not you, I don't remember. And another protein you might want to produce, it's the enzymes that are needed to digest lactose, okay? Now, this regulation occurs in a similar but different way. So you want to digest lactose. And typically you don't produce these proteins, okay? Or actually, I mean, when I say you don't produce, you produce a very, very low amount because there is always like these binds and binds, so there is always some very small rate. So transcription is blocked. There is no, you don't produce the enzymes needed, or you produce at a very low level needed to produce lactose. Okay. Now, suppose that lactose arrive, you drink your cup of milk. What happens is that now the digestion of lactose produce some molecule, which is an inducer, which binds to this protein here and release this. Okay? So what happens is that now you start producing these enzymes needed and other machinery needed to digest lactose. Okay? Now, this thing, to answer your question, this protein here and this protein here are two different proteins. Okay? So these names here corresponds to enzymes or other proteins that are needed to produce arginine. These are needed to transport and digest lactose, so there are different parts. And this is specific. So all these things here are specific, these things is specific.
So the repressors are
two different proteins.
two different.
Yes. The sequence here, it's different from the sequence here, and this protein here recognizes this sequence. And now you can imagine that the moment you have this, you can imagine that you can do all sorts of games, right? So what you can do is that you can have, well, you have something that regulates two different things at the same time, but then the second one also needs another condition. So if you want, it's not hard for you to think that you can build all sorts of logical gates with this, right? So given this condition, I'm going to do that, and then given this other condition, I'm going to do this something else. Okay? Yes?
RNA polymers are in different types, or it is one
For
type?
what we want to say is all the same. It's just the same machinery that moves.
How RNA know what to copy? We want this.
So there is a rate at which RNA polymerase binds here, and then when it binds, it produces it.
It binds the specific bar.
No, this one, it's sort of generic. Right? This one, but this one is specific. So yeah. So the rate you have, basically, you can have things that are produced in absence of this, that are always on. Okay. Things that are always on are still produced at a different rate because this binds with a different rate. But those are things you don't control. So you always produce them. Right? So you always produce them at some rate. Okay? Then you have transcription factors, you have regulation, okay? Transcription and regulation if you want, and therefore you are able to produce some proteins or not produce it only in determinate conditions. Okay?
To imagine it better, so the DNA have like multiple RNA polymers reducing different structure.
Yeah, at a given time, you have DNA, you can imagine it as a long chain, and at a given time you will see RNA polymerase bounce to different places, yes. You don't have just one RNA polymerase, they are all equal or similar to each other, but there are many of them, right?
Bending in different parts.
Exactly,
This
exactly,
big
yes, exactly,
structure.
exactly, yes. Other questions? Yes.
How does the signal of, well, you just drank the glass of milk arise?
Okay. Not next slide, but this is, okay, I'll explain in a different way. So this is like what I explained for arginine, right? So in this case, you want to produce an amino acid, arginine. So this is costly, right? A cell needs to spend energy to produce it, and it needs it. It needs to build proteins. Okay? Now, you want to produce it only if you don't have enough around. Okay? If you don't have enough around, you want to produce it. And yes, I'll tell it later. So you want to produce it, and the way it does is, if there is no arginine around, you sort of produce all the machinery all the enzymes that are needed to catalyze the reaction to produce arginine. If there is arginine around, you block this. Okay? We're going to see how this leads to arrive to a steady state of arginine. Okay? Now, how does it work for lactose? Actually, this is like, well, if you discover it, if you discovered it in the 60s, you would have won the Nobel Prize. But the way it works for lactose it's a little bit more. And actually, the story of how they discovered it, it's beautiful. It's beautiful and super ingenious set of experiments that I don't have time to cover, but it's like really cool. So basically, the idea is the following. So you always produce, so you have basically the set of instructions to deal with lactose. It's called an operon, right? And basically are the instructions to produce different proteins. Now the main actors here, it's like a protein that is a transcription factor. So that binds to this, to DNA. So there is this thing here. And then there are three main proteins. Actually, we care only about two also because I don't remember this one. So one is a transporter. So it's something that a cell uses to take lactose from the environment. So lactose is the sugar, sorry, I didn't say that. Lactose is the sugar that is inside milk, okay? And in order to digest it, you have to break it into two, it's a disaccharide. You have to break it into monosaccharides. One is glucose and the other one is galactose. So lactose is made of these two things and you have to break this bond. Now, you need two main proteins. One is this protein here, which is a transporter. So takes lactose from outside the cell and brings it out inside. And the other one, it's really the enzyme that breaks this bond. Okay? Now, how the feedback works. So in absence of lactose, you produce this repressor, which is always bound to DNA. Okay? And so you don't produce this. Okay? So you have something that tells you don't produce this. Now, when it tells you don't produce this, you have to imagine that you always produce a little bit because this binds and unbinds. So sometimes RNA polymerase attach and arrive at the right moment and start producing it. But you have very little. Okay? So the cost for you is very small. You produce very little. Now, what happens is that you have very little, you have some transporters there. And now you drink your glass of milk, and then there is a lot of lactose around, okay? There is a lot of lactose around, and then this starts getting imported in the cell, okay, by the few transporters you had. And the few enzymes that you had start breaking it, right? So you start breaking it. And now when you start breaking it, you produce many other things in addition to glucose as galactose, and you produce in particular one molecule that basically binds to this like I, to this protein, and make it not being bound to DNA. And so then you start to produce a lot of this. You start to produce a lot of transporters, a lot of enzymes. You start to take glucose inside, lactose inside, and you produce all of them because you keep like adding lactose up to the moment where there is not lactose anymore. So there is not lactose anymore. So you don't have this red arrow. And then this one binds again, and you start producing it. Okay? Yes?
For people who are lactose intolerant,
yes.
The enzyme that is
you don't produce this enzyme, this lactase. So this is the same mechanisms that we have. So basically, I'm not an expert in lactose intolerant, but my understanding is we all have this, because all mammals, because we all drink milk when we're born. But then there is a moment where we stop regulating this, and we don't produce it anymore. So basically, when you drink a lactose-free milk, what you are drinking is something where the lactose is already broken into glucose and galactose. Okay?
So the problem isn't in the transporters, it's in the...
Is in the enzyme, yes.
That breaks down.
In fact, I think that when people that are lactose intolerant take the pill, there is a pill you can take, it's basically the enzyme to break it. If it was the transporter, it will not be a problem, right? You will just, I'm guessing, I'm not, No, it just passed, you eat, yeah, just, you don't digest it, yeah. I'm not sure though, okay. If you want, I can check. Other questions? Related or not related to lactose, yeah?
And when you don't have lactose anymore, it's the arginine that stops.
No, this is two different mechanisms. These are two independent mechanisms. this perhaps was not clear, but these two are two independent. So this guy here and this guy are completely different proteins. They have nothing to do with each other, okay? It's just an example of how regulation occurs, right? Here, you want to produce something, okay? And you have a protein which is usually is not doing anything, but then when there is the product accumulating, it binds and it stops. Here you have something which is always repressed unless a condition is met, okay? Yes, these are two examples. Yes, lactose is in the second case, yes.
In the second case, when a condition is met, the repressor like have a lifetime or half a lifetime to like again repress.
I mean, all of these are, we are gonna sort of see it, but no, I mean, all of these you can imagine that there is a rate, right? So there is a rate at which this inducer binds to this one and a rate at which it unbinds, right? So you have, we're gonna see that. And then this one, there is a rate at which it binds and a rate at which it unbinds, okay? So, yeah.
Depending on the concentration of the material.
Depending on the concentration and depending on chemical properties, yes. Okay?
I have another question. I'm not sure if I heard what the LAC-A does.
I don't remember. I have to check it. If you want, I can check it right now. Yes, the other question?
Large quantity of cells are doing this process simultaneously.
I mean, okay, so when you have multiple cells, all of them, they are doing it. For all the discussion that I'm gonna say that later, but for all the discussion I'm gonna have in this week, is gonna imagine that all the cells are doing exactly the same things. There will be lectures in 2 weeks that will study exactly the differences between individual cells. For me now, a cell, 2 cells or 1 big cell is the same. I'll forget about it. But in principle, you have also this difference, which is important.
The point of this question was if all of them at exactly the same time do the repression maybe, and the function of the products can lose as continuous or as a step.
Well, you can imagine that it's continuous because, I mean, it depends on the time scale, but you can imagine that it's continuous because, I mean, it's continuous, and also it's not something which is exactly on or off, right? This binds and unbinds, so on average, this is the effect, but you have differences between cells. You have fluctuations, and fluctuations are important, okay? So at the level of individual cells, these, for instance, when this can still bind or unbinds, and this can bind or unbinds, right? So it's going on, off, on, off, on, off. So it determines the rate, and how much is going on, off, it depends on the chemical properties, but determines differences across cells. That's a very good point. Yes?
So this is not deterministic, it's a dynamical system, there are bifurcations.
Yes. I mean, it's that, I mean, I'm not sure about the not deterministic, so it's true that it's not deterministic for the reason I told you. But even if you forget about this fluctuation, when it's deterministic, you still have bifurcations, etc. So, well, I'll check later. Okay, so this was the 4th point. So basically putting the points together, there are chemical reactions, and you can do many of them. So for instance, this is something that is a first step to digest lactose, which is that something that produces energy. This is something that is needed to produce amino acids, to use energy to produce something else. But all the reactions that occur because there are enzymes that are specific, and these specific enzymes are regulated by, and all the other processes that are needed, like the transporters, are regulated in a cell. So a cell can, in this way, have a feedback between what's going on around the cell, inside the cell, and what the cell is doing. Okay? Questions? Okay, so the other thing that a cell has to do, okay, this is I showed you, blah, blah, blah. Okay, the other thing that a cell has to do, and I will not cover this, is that a cell has to duplicate, right? So there is this fantastic quote from François Jacopo, which is the guy that together with Monod discovered the Lac-Operon, which is that the dream of every cell is to become two cells, okay? So what a cell has to do is that it has all this instruction in DNA, and at a certain point has to duplicate this DNA, and from one copy of the DNA, make two copies of the DNA, and then segregate and becomes two cells, okay? Now, as I mentioned in the question a couple of minutes ago, I will forget about this process because this is a process on how, exactly, that has to do with individual cells. And for me, I will forget about this. For me, whether you have two cells or you have one cell is the same. I will just look at the bulk, so all the cells together, but this is gonna be covered in two weeks, okay? Or part of this. Okay, so this is the summary of what I explained so far. So there are cells that perform reactions to get energy and produce new materials. There are enzymes that make this reaction occur in finite times and are specific. The instructions to build these enzymes are written on DNA. And cells take decisions by regulating which proteins express and how much. And cell replicate their instructions and becomes two cells. Okay? So these are the things you have to remember. Questions? Yes?
I don't know anything about biology, but like you have said, everything is stochastic up to encountering the cell and stuff. Can we think about this as almost deterministic, meaning that we have enough ribosomes and stuff to say that this is almost.
So this is a very good question. So all these processes are stochastic, right? So there is a rate at which, I started saying there is a rate, right? So there is a rate at which something binds and a rate at which something unbinds. There is a rate at which the mRNA is transcribed, is translated, so everything is stochastic. So this stochasticity is important. Is important both, if you look at a single cell, there are no two single cells that are equal, and the fact that they are not equal matters. What we are gonna do this week is to forget about it. So to say, I look at the average. With the idea that, and I'll try to be more specific either today or tomorrow, with the idea that we are looking at what is called bulk growth. So we are not looking at a single cell under the microscope and looking how it divides, but we have a big flask with many cells that are growing, and we look at the population average. Now, going from this, if you want, ensemble average, population average, ensemble average, and time average, it's tricky, it's not obvious. It will be covered in 2 weeks by Relamir, but let's say, as a first approximation, we'll forget about stochasticity, okay? Other questions? Yeah? Yes, it's exactly like that, yes. So it's exactly like a two-state random process where you have a rate of binding and a rate of unbinding. It's exactly the same thing. Okay. Now, this was the simplified version, what I told you. Now, the reality is way, way more complex. Now, you know these simple pictures, so we can go back to the comfort of a nutshell. You know that these are the main components, but now, if you look at a cell, even a simple cell like Escherichia coli, which is a very lab-adapted, simple, streamlined bacterium, life is much more complicated, and you have to put all these things together. So, for instance, this map are all the metabolic pathways of E. coli. So, all the things in green are all the arrows are reactions that are performed by E. coli. Okay? Can you see the green? Yes. And so they are connected, and you can see that they perform many reactions, and you can imagine that each reaction corresponds to an enzyme that can catalyze this reaction, and the expression of this enzyme is regulated. Yes?
Is this all the possible reaction or all the reaction in the same time?
No, all the possible reactions.
All the possible.
Yes. Yes. But many of them are performed at the same time. Okay? But this is all the possible reactions. Now, as we said, it might perform some, not others, depending on the condition. But it has this is the way this is built, is looking at the genome. So these are the things that a cell is able to do. Then there might be things that it does all the time, and other things that it does very, very rarely in extreme environments. Okay?
This is for a specific type of
It's for Escherichia coli, which is a bug that lives in the guts of mammals, of cows, and it's sort of the standard model organisms for bacteria. Okay? So there are many things. Now, the one in red is what is called central metabolism, and you can zoom in, right? This is like sort of a part of the metabolism. You can zoom in, and if you zoom in, this is like regulation of the central metabolism. So you have all the one in yellow are like molecules that a cell can transform. So for instance, this is fructose, this is CO2, this is water, okay? And what a cell can do is that can transport things that are outside inside, and it can tune how much they transport it, okay? And then you have in blue the reactions, the same that appeared before. So you see that you have this cycle, is this cycle here, okay? So you have in blue these reactions, and then what you have is that you have regulation. So in red and green, you have activation and inhibition of an enzyme depending on the presence of some molecule, right? So as I showed you that lactose inhibits the production of that enzymes, you should be able to see that arrow, red arrow somewhere, but I think I will not find it because I don't know if it is in the lactose. It's here, lactose it's here, so it should be somewhere. Well, there will be some arrow pointing somewhere here at some point. Anyway, so the point here is you see that you have this regulation, right? And what I described is this little part here, okay? And you have all this rest. And so you co-regulate everything, and this, remember, is not everything that a cell does. This is just this red part here. Okay? And now you can even zoom more if you like more details. And this is glycolysis, so is the way you break out glucose to make energy. And each of these, it's like a set of chemical reactions which depends on some enzymes, have some activation rate, consume some amount of energy, needs some products, we need some energy or produce energy, and so on and so forth. Okay? So if we discard our nutshell where we put all our physiology, reality is way, way more complicated, and there are a lot of details that we didn't discuss. Okay? On the other hand, sort of the 5 general facts that I told you are behind each of these little details, or most of each of these little details. Okay, questions? Yes?
Regulation, this example for regulation is the combination of more than of many pathways.
Yes.
Like lactose in this case. And
Yes.
another process.
Yes, so basically here it tells you how you take different, in this case mostly sugars and other basic substances, and you break them, for instance, to produce energy, okay? Now, all these reactions which are in blue depends on other molecules, right? For instance, this thing here, which I don't know what it is, stops this reaction here, okay? And so you have these mechanisms where basically, depending on what is the internal and external, the internal state of the cell, so the amount that you have of each of these orange dots, you can make one reaction be faster or slower, okay? Occur more rapidly or less rapidly because you're gonna produce more or less enzymes, and so you change this level here, right? You change how much you produce, going up and down. Other questions? Yes.
Is some method to break these processes of general regulation, maybe another bacteria or something?
So the way, I interpret your question as how was this discovered? How?
Yes, if you can break the process in all the processes that are immersed in.
So I'll answer the question, how is this discovered, and then you tell me if I answer your question, okay? So the way this is built, was built over decades, is that you basically identify, you understand that there are chemical reactions. You identify what are the enzymes that are needed for these reactions, and you identify where they are on the genome. And then what you can do, for instance, is to construct a cell that doesn't have these instructions, okay? So you can play with the instructions. You say, this instruction now is not there. Or this instruction is always off. Or this instruction is on if I do this, right? And so by doing that, you can say, if I put lactose, then this is not produced, okay? And so on and so forth. And so you can sort of build this map, but, right? You can imagine that it's very tedious. Other questions? So if you want, in molecular biology, one paper is one error, okay? Other questions? Yes.
So the position of each red dot just tells us, or the position as the arrows just tells us how each, say, protein or enzyme is constructed. So that's what the arrow.
So the arrow, it tells you how it's regulated, right? So if you see an arrow here, it means that the presence of this binds, for instance, to some transcription factor that then blocks the production of the enzyme that is needed for this reaction in blue, okay? So if you have a lot of this reaction is gonna be slower, okay? If this is not there, that reaction might be faster.
Another question, is this not supposed to be readable, or?
My point is also to show you how readable, let's say, there is a meta message here that, of course, it's extremely I strongly simplified, but when you put everything together, it's extremely complicated, okay? And the reason why I'm showing this is because then we're gonna go back to write sort of simplified models, and it's important to have this in mind, right? That on one end, reality is very complicated, and on the other end, it's very surprising that simplified model works so well. Okay, so keep this in mind, right? You don't have to remember it. You have to remember the feeling that you have looking at this. Okay?
So when we simplify models, do we get results that are aligned with this general method?
Let's ask this question in a few days, okay? Other questions? Okay.
Can we ever be sure that we are not missing any apples?
No.
Maybe for this system, which is very good.
Let's say, if your question is, it becomes almost a philosophical question, right? Can we ever be sure that whatever we know it's enough? The answer is no. Now, this is a very well-characterized system, so most of what is to be known is known, but there are many unknown things, okay? I don't know if I answered, but. Okay, so it's time for me to stop. I just want to tell you one last thing, which is also a question for you, okay? Okay. So, okay. So how do you do, let's say, experiment related to microbes which are alive? What you do is that you have to be able to grow them, okay? So what you have is that you have some I'll go fast now, and then tomorrow I'll start from this slide again. But you take bacteria, let's say E. coli, you put it in a flask with some nutrients, let's say glucose, you shake it, and then you make sure that you shake it because you want oxygen to be there, and then you have it growing, okay? And so if you look how many bacteria you have, and for instance, you can look how turbid your culture is. What you see is that at the beginning they don't grow, then they start grow, and then they stop growing. Okay? And depending on the conditions, sometimes they go faster, sometimes they go slower. Depending how much nutrients you put, sometimes they go up, sometimes they grow less. Okay? Now, you can imagine that you do this, and then you can take your culture at any point in time, right? You can take it here, you can take it here, you can take it here, you can take whenever you want. And what you can do is that you can measure something. What you can measure? Well, for instance, you can measure how many bacteria there are, right? Or you can take them and look it under the microscope and look what fantastic shape they have, how big they are. Or what you can do is that you can do biochemistry, right? You can measure how many proteins there are inside these cells, what type of proteins there are, which are the proteins that they express, how much DNA they have. You can sequence DNA, you see how different it is, right? So this is in general what people do if you enter a microbiology lab. What people are doing all the time is growing bacteria and measuring stuff, okay? Now, the question that I have for you is, suppose that we have to study physiology. So we want to study what are the activity that a cell has to do that take place to be alive. So my question for tomorrow is, along this time, right, when is easier to study physiology? So when, and this question it's like fuzzy enough on purpose, but if you want to study these properties, when you want to collect your data, on which point of your curve you want to collect your data, okay? This is the question for tomorrow. And then tomorrow we'll start again from this. Huh? Whatever you want. You just want to see what a cell does to be alive, okay? Questions? Okay, go and digest. No, sorry. Very important. I already failed my first task. You have to sign this. Okay? So come here and sign it. Hey. Here, no, this is...